My name is Mariah Hassert, and I'm a postdoc at the University of Iowa. And today I'm going to describe an exciting new article from the Harty lab entitled Regenerating Murine CD8 Lung Tissue Resident Memory T Cells After Targeted Radiation Exposure. The use of radiation is substantially increasing. In developed countries, up to 50% of our radiation exposure comes from medical procedures, and nuclear energy ranks as the second largest global source for low-carbon electricity. With this increase in usage comes an increased potential for accidental exposures. Despite this, effective medical countermeasures to deal with the negative effects of radiation exposure are abysmal. Radiation can be used in a medical setting to target tumors, which can have a high proliferative capacity. The radiation exposure causes DNA damage to the cells, which results in their death. As cells that can proliferate rapidly, CD8 T cells can also be susceptible to this death. It has been appreciated that naive T cells are susceptible to radiation-induced depletion. However, over time, their numbers ultimately recover. The effect of radiation on memory T cells is comparatively understudied. In a PNAS article published in 2023, our collaborators in the Badavinak lab described the long-term effects of total body irradiation on the circulating memory T cells. They found that, like naive T cells, circulating memory T cells are susceptible to radiation-induced death. However, they do not recover with time. In this paper, we looked at the susceptibility of tissue-resident memory CD8 T cells, or TRM. Rather than circulating throughout the body, this T cell subset remains lodged within tissues poised to respond locally. We utilized the murine influenza A virus infection system. This infection generates both circulating memory T cells and lung-resident T cells. It is the TRM which are critical to protection from heterosubtypic influenza infection. To study the effects of radiation exposure specifically to the tissue, we utilized a small animal radiation research platform, or SARP, which permits the targeting of radiation exclusively to the thorax using CT image guidance. Following thorax-targeted irradiation, lung TRM are numerically reduced, as you can see in this top panel, and they did not numerically recover over time, which resulted in compromised heterosubtypic immunity to influenza infection. In contrast to lung TRM, the circulating memory T cell compartment was left fully intact, which you can see on the bottom panel. We hypothesized that we could capitalize on these circulating memory T cells as a source with which to regenerate lung TRM. To test this hypothesis, we boosted these mice with a different strain of influenza and found a significant increase not only in circulating memory T cells, but also lung-resident memory T cells, indicating that there was an intervention that could be made to recover these cells. Importantly, we could regenerate these cells not only with a localized influenza infection, but also with a peripheral infection with recombinant Listeria monocytogenes, as well as a more clinically relevant mRNA vaccine. These recovered lung TRM were generated from an influx of circulating memory T cells and subsequent conversion to TRM, rather than the proliferation of the relatively few TRM that survived radiation exposure. These findings indicate that radiation exposure can have significant consequences to the tissue-resident memory T cell compartment that result in compromised immunity, but does offer hope that in the context of targeted radiation exposure, for example during radiotherapy for the treatment of tumors, that medical intervention can be made to restore this critical cell population.